Okay, well, welcome back. This is the fourth lecture of E443, 543, talking about virtual tissue modeling methodologies. Today, we're going to start out by talking about cell growth and division. We're going to be doing a fair amount about plotting and how to do display of information inside of CompuCell. We're going to talk about tracking fields and cell sorting, at least if people are interested in going over some of that. And I need to remind you that this lecture is live streamed and recorded, so please be aware of that. So I'd like to start by just quickly going around the room and having people review where they are with projects. I'm going to ask you to do a more formal presentation next week. And I also handed out a homework assignment. And so starting next week, I'm going to ask people on a weekly basis to give themselves a little mini assignment, which is what am I going to do for the next week on my project? And you're going to have a second assignment, which is answering that question. So there are going to be two homework assignment problems week by week. The first one of which is, what did I do last week? And the second of which is, what am I planning to do for the following week? And you'll get 10 points for each one if you do a reasonable job answering them. So this idea is to help keep people on target and motivated. For the week after, for this following week, I'm going to ask you to do a little bit more detail thinking about the problems. I know I talked to several people one-on-one in the past few days about projects. People seem to have some very good project ideas. Some people are grouping together in teams. That's great. As I've said before, people come from very different backgrounds and skill sets. And so working with people with complementary expertise is a great idea. Then I'm going to review some of the things we did in class last time. Again, if these reviews are too fast, you're welcome to say, slow down and explain something the way you just did. That's useful. If you say, boy, we've seen that already, you can put that in the chat to me or say so out loud. I'm not shy about being told I'm going too slowly or too fast. And then we're going to talk about having cell volumes change. We're going to talk about this pressure and surface tension and how to display them. And then depending on how far we get, I thought I might talk a little bit about some review of problems from the homework 2, which we could skip or go through relatively quickly. I was going to potentially review that cell sorting problem that people were asking about just now, and then talking about cell division, a little bit about the cell cycle and the biology, and then how to implement cell division in CompuCell, and then possibly some exercises. If we don't get to cell division this week, we'll also do division next week. So maybe to begin with, I'd like to get quick updates on your projects. It doesn't have to be very long, but at least so that I know where people stand. So I guess we'll go around the room. Joel, you're at the top left for me, so why don't we start with you?
Right. I talked with Dr. Glazier in 101, and we noticed that I have to get a bunch of missing data or how to implement some data in my project. So one of the things that I have to do, or that I started, was looking up all the data that I actually have in the model presented in the paper, and whatever I don't have, I will look up or email Gary, that is one of the writers of the paper, to get more information. And another thing that I was incentivized was to put it in a manner using the workflow we read on week 1, to keep things like very well organized and on a proper way, since it seems, as we read, it seems to be like a good model to follow. So that's the plan that was trying to execute. I was not fully done with it, but it will be something that I will keep doing across next week.
Okay, thank you. Connor.
So at the end of last week, I met with Professor Glazier to talk through the multicellularity paper I was looking to replicate some results from. And we set up a plan of kind of steps to go through, and which will probably end up being pretty similar to what I choose to be weekly assignments at each point. And we also went through some code that I still need to sit down and process through a little bit more thoroughly, but to get some basic stuff for chemotaxis set up. And I don't think he's here right now, but Andrew decided to join me in trying to replicate that paper. So I'm going to meet with him probably tomorrow or the next day so we can coordinate on that. The one thing I so far I have, we emailed one of the authors about chemotaxis and no response yet. I'm thinking that like in a few more days I'll follow up if I don't hear anything. Don't want to bug them too much, but yeah.
Well, so we now have two people who've had the issue that the published paper isn't reproducible because it doesn't have enough information in it. It's a little embarrassing that one of those papers uses CompuCell, because one of the purposes of CompuCell was to make it easy to publish models in a reproducible form. So it's a little bit disappointing in a way, but we'll do our best to get that working. I think even if we can't explicitly reproduce all of that paper, I think there's quite a bit that we could do conceptually from that paper, even if we don't have the details. Alex.
Yeah, I met with TJ this morning, and we discussed several new feature enhancements and like tech debt projects that can be done to CompuCell. So I need to dig into those. He gave me like 4 ideas, so I need to dig into them to pick which one I'm going to do.
That's great. It's always nice if people want to work on core code. It's not that often that people do, and it's very valuable because it's an open source project. If your core code finds its way into the code base, then lots of people benefit. So it's a nice way to contribute if you're willing to do it. Appreciate that. Jackson.
So my initial ideas didn't seem to really work out. So I've been looking to do just replicate the results of a paper. And I was thinking about doing just the first computational modeling of angiogenesis paper.
Okay, so we should probably meet and talk that one over and see what's possible.
Okay.
Because I think it's important to think these through. It's a lot of your time, and so you want to make sure that you're targeting on something that's going to be interesting and productive. So let's set up a meeting for that.
Okay.
Josh.
Hey, so Dr. Simler emailed me back and he sent me all of the code for the project. I haven't tried to run it yet, but I think it's moving in a good direction.
So depending on the year that was written, it won't run.
Mm-hmm.
You could do two things. You could either load an old version of CompuCell, the one that it was written for, or potentially you could we could show you how to upgrade it. So that's possible. Great.
Okay.
That's good. Let's see. Anyone from Purdue want to say anything about projects or ideas? No, it's okay. we're interested, if people at Purdue have suggestions for things that should be modeled, like wound healing in tail bud, that could be suggested as a project, by the way. So don't worry about it. Or calcium waves, these kinds of things. These are all possible. You have an audience of people looking for projects. Okay, Andrew.
Hello? Can you hear me?
Yeah, we can hear you.
Okay. Sorry, I just entered the room. I don't know exactly what's going on right now.
So I think Connor said that you were going to team up. Is that right?
Yes. Yes, we're working on that multicellular paper, replicating it.
Okay, so I hope that that's worthwhile.
Mhm.
And again, it's not required that people team up, but if somebody doesn't have a project, I think Jackson, if you say that if somebody says that they or doesn't have a project idea, you might talk to somebody else like Joel, who does have a project idea, and see whether working together on it might be reasonable. There's no need for people to feel they have to do a project on their own. So I encourage people to take advantage of that. All right. So I want to review some of the key ideas from last week. One of the things that we had that I think is so important, I'm going to keep coming back to it, is this question of what is a reasonable set of parameters. And reasonable will depend fundamentally on the biology and the physics of the problem you were trying to describe. But one of the things that we're going to run into frequently is there is this parameter called temperature, which describes fluctuations in our system. And that parameter comes out of the air to some extent, showing up in this energy acceptance functional in the statistical properties of the way cells move. And it really represents biologically the membrane fluctuations of cells due to cytoskeletal activity. And the question people continually will ask is how do you know what it should be? And I want to come back to the idea that what matters is the ratio of typical energy fluctuations. And here, energy is written with a curly H instead of an E. As I mentioned, that will oscillate depending on who wrote the slide originally. And what will matter is the ratio of that energy change, that energy scale to that T parameter. When that energy scale is too small, delta H over T is a large number. If delta H over T is, I better get my signs right. If delta H over T is very small, then almost every voxel update is accepted. And that means that the energy functionals that you're using to control the system don't do much controlling. And you'll have situations like the one we see here in the second to left, which is a cell that's beginning to fall apart. You have lots of separated voxels. If delta H over T is very large, then almost no updates are accepted. Things don't move, and you have what we call freezing, which is in fact analogous to freezing of water. And you'll get a situation like the one in the far right with the green cells where the boundaries line up with the lattice and don't move very much. There's a large range, however, of fluctuations and energies where the typical fluctuation is smaller than the typical energy, but not by very much. And in those situations, you'll get something that looks, what we'd say, better behaved, like the cell crawling here on the left. It's moving in a fluid way. The boundary is rough, but not falling apart. Or here in the second to the right, where the cells are fluctuating and rearranging, but their boundaries are well behaved. And so we should be thinking about that. There are times when cells do fall apart. There are times when cells do become rigid. If you're dealing with plants, the cell walls are almost always rigid. Bacteria, they're rigid. And there are cells that there are situations like the true slime molds where there are no boundaries between cells. So both of those things do happen. But in general, the situation on the left or second to right are the ones that are going to be most common biologically. And so we're going to want to try to adjust things to do that. And we have some basic ideas that if the fluctuations are too small compared to the effective energy, then the cells won't move. We'll get cell walls lining up with the lattice, and we'll get corners, sharp corners on our cells. If the fluctuations are too big compared to the effective energies, the cells will split into multiple domains. You'll have lots of stray voxels, and the cell boundaries will crumple, as you see here on the rightmost picture with the blue. And I think the homeworks that you did last week and this week explored that in some detail. And one could do a lot more. I had originally had a lot more exercises related to that. If people are interested, I'll outline some of the things you could check. Again, we can understand that by looking at that acceptance function. Remember that if a voxel copy reduces the pattern energy, we always make it. The question is what happens if we try to undo the thing we just did. The energy to do it and the energy to undo it are minus each other. So if going forward is favorable, going backwards will be unfavorable with the same energy. And the probability with which we accept that will be an exponentially decreasing function of delta H divided by T. And so here we've shown an example where suppose that the energy cost here is indicated by this red arrow. If we have a T here of some value, which is the red one, we have a probability of going backwards of about 1/10. If we make the temperature, that fluctuation amplitude, 10 times smaller, this exponential moves far to the left, and the probability of going backwards is essentially 0. And so changing this fluctuation amplitude has a very strong effect on how likely it is to go backwards after you've made a favorable step. We did a quite extensive exploration in class and also in the homework of some of the cell medium and cell-cell contact energy effects. We looked at how higher cell medium contact energy leads to rounding up of aggregates. The higher the cell medium contact energy, the smoother the aggregate, the faster the rounding. If the cell medium contact energy is smaller, the aggregate gets looser. If the cell medium contact energy gets less than about a half of the cell-cell contact energy, then the cells will actually disperse into the medium and spread out. If we have small cell-cell contact energy, we'll get crumpled intercell boundaries, which we saw in the previous picture. If we have small cell medium contact energy, we'll get also rough cell shapes. And so depending on whether our cells in fact are smooth and polygonal or rough and full of protrusions, we'll pick different contact energies. We talked a lot also about the code structure of CompuCell. Particularly, we started working with the Python part of CompuCell. And the basic computational architecture is called a plugin. The plugin is a set of classes which we're going to use to define things that we're going to do to our cells. There are a number of functions within the class. There's the init function, which we're going to use today. We don't use it that much, actually, but we'll use it today, which is called before the first time step in CompuCell. There's the start function, which is called once at the first time step. The step function, which is called periodically every MCS number of steps. The finish function, which is called when the system exhausts its number of Monte Carlo steps normally, and now relatively recently this on stop, which is called when you hit the stop button in player. And these can be used, finish and on stop can be used if you want to save the configuration of your simulation when you're exiting. The key thing here is that frequency at which you call simulations. You have self frequency up here equals 1. That is going to be defining how often these are called. Should have been called period rather than frequency. Another thing that we talked about and we'll work with fairly extensively, and you've had experience now also in the homeworks, is this concept of cell objects, computational cell objects, that is. CompuCell has this idea of a cell, which is one of its main concepts, and the series of attributes that the cell has. And one of the things that you can do in Python, and you'll hear me recommend it frequently, is there's a Python function called dir directory. And if you do a print parenthesis dir parenthesis object name close parenthesis, you'll get a list of all of the attributes of the object, all of the methods that are available for the object. And that can be very helpful in helping you understand what you can do with an object if you forget. Cell attributes, we have a nice menu in Twidit that lists a bunch of them. I'm not sure that it lists all of them, but it lists most of them. And we can look at this list. What's the type of the cell? Where is the center of mass of the cell? What is cluster ID? We haven't gotten to yet. That's for compartmental cells. We'll do that later in the class. What is the identity of the cell? It's an index. Inertia tensor describes the shape of the cell. Lambda surface and lambda volume we know about. Surface area and target surface area, target volume and volume we know about. Things that have been added relatively recently, so they're not showing up in that list, are the cell pressure, which we'll work with today, and the cell surface tension, which we'll also work with today. Something that we're going to do a lot, and you've already done it in your homeworks, is iterating over things. Classic iterator that we'll use again and again is over all the cells in the simulation. Those are kept in an object called self.cell_list. You also have the option of iterating over all cells of a given type, all lattice pixels, all boundaries of a cell, and in the homework that was asked about, all neighbors of a cell. And that neighbor iterator has slightly unusual structure, and we'll play with that later. And actually, the way I've got the problem solved in my slides, it may not be the best way to do it. It may not be the way that it would be natural to do it, and we can do that out together on the fly if people like. And we're going to do this kind of iteration all the time. Most of these iterators are very well behaved. As I mentioned, the neighbor iterator has one peculiarity because it doesn't return just the cell neighbor returns a more complex object. Iterating over all lattice pixels is unproblematic but slow. So it's something you would like to avoid doing if you can. And we'll talk later in the class about how to avoid that. Something that you may want to do, and that is a slight peculiarity in CompuCell, is that you might want to reference cell types. And when you create the XML, you remember you always have to list the types of the cells. That's one of the first things you do in Wizard, is list all the cell types. Those type names get turned into Python in a slightly funny way, whether Python, normally XML doesn't care about capitalization very much, and the Python does care about capitalization. In this particular case, it's backwards. And when you reference a cell type inside of a Python, you have to use the type name in all caps. So if the type of your cell was CELL, all in lowercase, the reference to that cell type in Python would be self dot capital C, capital E, capital L, capital L. And so that's a little bit of a trap there for people to get confused about. Once you know that that's the case, you can do iterators like for cell and self.cell list by type. And you could say self.dark cell, comma, self.light cell, self.whatever cell names you have. And so this iterator by cell type is useful. One of the things that you may run into when you use these lists is that some of these cell lists are in fact lists and some aren't. And so there may be, typically the things we do won't matter, won't care about that. And the list operators like length work on them. But occasionally you'll run into one function that operates on lists that will have a problem with them. We'll talk about those when we come to it. The next thing that we talked about last week, and I think people had some issues or questions about, was creating scientific plots. We'll do that plenty today. There are always 3 steps in creating a plot. The first thing is that you have to create a window in Player where you're going to display the plot. And that uses the function add new plot window. And you have to give that plot window a handle, which is here given self.plot_win. That name plot_win is user defined. It could be self.aardvark equals self.add new plot window. But you do have to remember it because you have to have that name be unique and you have to know how to reference it. So you have to make that into something you can keep track of. And it's pretty easy if you create 5 plot windows to forget which one is which and accidentally try to create 2 plot windows with the same handle. That won't work. Or accidentally add your data to the wrong plot window. Those happen. Some of those will throw an error. Some of them will simply silently do the wrong thing or do nothing. And so that's something you have to keep track of by hand. Title equals here average volume and surface. For reasons that I do not understand, you can't reuse the title. If you try to create 2 plot windows with identical titles, it'll throw an error. And I assume that's because the plot windows are actually stored as dictionaries, and there's a dictionary key, which is the title of the plot window. Because it doesn't make a lot of sense to me otherwise. I actually don't know why that is. But
So, oh, sorry.
Go ahead.
Is it that you can't, like, so if I wanted to label on my x-axis, like it says like Monte Carlo steps, does that mean I can't, is it just that you can't use the two same axes grouped together twice, or is it that you actually can't use like a label twice?
The axis labels are fine. You can
Okay,
reuse
okay.
those. It's the title of the plot window that has to be different. And I genuinely don't understand. I understand why the handle for the plot window has to be different, but I don't actually understand why. I think it's because add new plot window actually creates a dictionary entry somewhere, which is keyed by the title. And so if you try to use the title over again, you're trying to create two dictionary elements with the same key. But I don't know. I don't know why the function has this form. But you do have to have a different title. We should probably emphasize that because that is a little bit of a trap. Typically, you don't want to plot the same thing twice, and so it's not that common that you do it. But if you're boilerplating and you don't think to change the title, you can have an issue. And there are a variety of options like scale type equals linear or logarithmic. We were having some trouble with some of the logarithmic plotting, scaling. I don't know if that's fixed or not. You can have grid lines, and there are a variety of other options that we can look up. But we could create as many plot windows as we like, and that's something we will typically do. Once we have a plot window, we have to add data series to it. Again, you have to remember to use the correct handle for the plot window. So if you have plot win 1 and plot win 2, you have to remember which one to put it in. Then within a single plot window, you have to have a unique name for the data series for each data series you're going to put in the window. You can reuse a name between plot windows. So I could have mvol in plot window 1 and mvol in plot window 2 that would be different. That's allowed. The typical things we're going to set as options are whether we have lines or dots. There are other options. What color they're going to be and how big they're going to be. Once we've created a data series, this is typically done, plot window and data series are typically done in the start function. Occasionally we'll create a plot window and a step function because we'll pop a plot up if something happens during the simulation. Adding to the data series, we have to remember what the handle for the plot window was. We have to remember the name of the data series. So we do wind up having to thumb back and forth to the place we created it. If our steppables are small, that's no problem. If we've got big steppables, sometimes you have to do a little bit of thumbing around to find them. If you try to put a variable into a plot window where the variable doesn't exist, it doesn't throw an error. It doesn't plot, but doesn't throw an error. So that's something to be aware of. Are there any questions about plotting? I know there were a few emails to me about plots.
My primary issue of plotting was less about the actual structure of how to do it, more with the specific case. I ended up going through some of the demos and I made some progress, but there was still definitely an error in how I was structuring my code.
Okay, well, if we can let's go forward a little bit, and then if you like, we can do some debugging together if people are interested. That would be okay. Is there something you wanted to discuss now about that?
I can bring it up when it's more topical to where we're at. Yeah.
But please, if you're having an issue with it, almost certainly everybody else is too. So the next thing that I want to do is to start an exercise which is about manipulating cell properties on a per cell basis. And to begin with, we started out by defining most of our cell properties in XML. And that XML is very convenient. And we can actually change XML parameters from Python. Although it's not the most convenient thing in the world, it's not terrible. However, it's not as convenient. And if we want to have things like, what we're going to focus on in this class is going to be cell growth. And if we want to be able to have cells individually grow, that means that the target volume of the cell is going to depend on which cell it is, not what the type of the cell is. And so if we want to be able to change the target volume or target surface area of a cell individually, we have to define it cell by cell basis, not by type. And so I want to go over the process of doing that. And so I'd like people to get CompuCell running, whether it's on the desktop or on NanoHub. And I do have versions of this code available in the student materials. There's a student materials exercise for a lecture 4 demos folder if you want it. But this is actually pretty straightforward. It shouldn't take more than a minute or two to do it using Wizard. So what I'd like people to do is generate a base simulation. First, take a minute or two to get CompuCell running. And then I would like to first generate a simple simulation. And what I want is, we'll do it in 3D. We've been doing things in 2D, but we can do it in 3D. Create a 100 cubed lattice, single cell type, we'll call it cell 1, with surface and volume constraint, and use the blob initializer, not the uniform initializer, and make a sphere of cells centered on the center of the simulation with a radius of 20, make the individual cells 5 by 5 by 5 without a gap. And then you can set the target volume and target surface of the cell to be 125 and 2. And the lambda volume, target volume and target surface both to be 125, and the lambda volume and lambda surface to be 2. And then once you've done that, generate a pair of plot windows, one that plots the cell volume, and one that plots the cell surface. And I think actually in my simulation, I put them in one, I put those data series into one window rather than two. And to begin with, why don't you plot the individual volumes and surface for each cell versus time. And then after you've got that working, add a little bit of code to plot the mean cell volume and surface versus time, and maybe make that a line overlaid on the individual cell volumes and surface. And here's one of these comments that I made. If you want to know how many cells you have in the simulation, one way to get that number is to use the function len, which tells you how many elements there are in a list. If you do len of self.cell_list, it'll tell you how many cells there are in the simulation. So is that a sensible exercise? If you've already done all of it and you have it running already, that's fine. You might have done it in homework. If not, why don't you give it a shot and see how it goes? So I'll give people a minute or two to try that out. And if somebody has a question, please feel free to ask.
Do you want like a separate line for each cell? How do you want us to?
You mean how to plot the cell volumes and surfaces? So, yeah. So no, I would use dots. So for each, so what I would do, you could do it a variety of ways. What I would do is at each time step, I would iterate over all cells. and I would add 1 point, which is the current volume and the current surface of that cell to the plot. So you'll get essentially a dot plot, which will give you a cloud of points. If all the cells had identical volume, then you just get a single point because you'd be overlaying it a lot. If there was a lot of dispersal in the volumes, you'd get a big range. So it gives you a sense of what the range of volumes is. And that's something which is useful if you were to adjust lambda volume. When you make lambda volume very large, it forces all the volumes to be close to their target, and so the dispersal would go down. If you make lambda volume small, the variance will go up, so you can see it that way. The one problem with that, with the plots, is that if you have a lot of points in a plot, because every time it adds a time step to the plot, it redraws the whole plot. If you get millions of points in your plot, it will slow down the simulation. But for our purposes, the simulation will run so fast we won't notice that.
Can you rename files in Twedit?
Yes, there's a save as option which will rename the file. And you definitely want to use the save as, because it's CompuCell simulation is not a single simulation file. It's a cluster of files in the directory architecture. And so you definitely want to use that renaming function, because that will handle all of that renaming. If you try to do it by hand, it's likely to get a path error. I just walked through the exercise, and almost all the defaults will be correct if you use Wizard to set things up. The one thing you'll have to edit two things you'll have to edit by hand are the target volume, target surface area, and the size of the initial cells in the blob initializer.
I think I have it for the averages, maybe. I literally just got it to work, so.
Okay, that's fine. Let's see.
All right. At the very least, I didn't change them to lines, but I have the 2 dots happening here.
Okay, that looks good. How did you do? Can we see your code to see how you did it?
Sure, yeah, let me go ahead and switch to my other screen. Here we go. So I went ahead and go ahead and set things up regularly. So I set up average volume here, average surface, set the colors to be red and green, and the sizes to be the same dots. And then for the step, I set both of these to 0. And then for each cell in the list, I use the accumulator to add it. And then after I summed all that up, I divided it by the length of the cell list and then plotted it.
That's how I did it.
Cool.
Did anybody else have a different solution? Juliano?
I do. So, let me show the code. Well, first let's show CompuCell. Correct, CompuCell. I have 2 running there. So I have the 2 plots, one for surface, one for volume, with all of the cells individually as red dots and the line, the mean. And the only real difference, where's the controls? The only real difference from your code is how I did the mean, which something that James Lucas said once he's part of the lab is not to do things by hand when they exist by somebody that audited the methods. So I went ahead and from Numpy, I imported mean. And
Can you possibly zoom in a little bit, because the
sure.
font is small?
Yeah. So from NumPy, I imported mean since I only need one function from NumPy. Usually I would import everything. And then in the step, I have two lists, one for volume, one for surface. And then for each cell, I'm appending the volume to the volume list and appending the surface to the surface list. And then I'm using NumPy's min function to calculate the mean. And the point is to just not do it by hand, because I can make a mistake. If NumPy makes a mistake, people will know. If I make a mistake, nobody will know, not even me, probably.
So that's a good point. I think the one thing to think about here, I usually often will automatically import NumPy. Does everybody know what NumPy is? Is there anyone who doesn't know what NumPy is? Maybe it's a better way to say it. So I know some people, we have some people who are not big Pythonic experts. But NumPy is a standard utility for doing numerical functions. And typically I will put at the beginning of almost all code I write, import NumPy as NP. And then you could use all of the NumPy functions as NP dot something or other. There we go. And in that case, those become available. If all we're going to do is get the average, the way Connor and I did it makes sense. But if you wanted to plot the mean and the standard deviation, for example, then you might very well want to do what Giuliano did, which is have a list of all of the volumes. And then you would be able to operate on that with any functions that NumPy offered. In terms of iterability, this is a place where that lack of iterability is something we ran into. You might think that if we could do something like apply a function, land on the self.cell list, that the list of cell volumes ought to be a list. But they're not. The cell volumes are attached to the individual cells. And so we have to build that list of cell volumes ourselves if we want to operate on it. And so that's the kind of place where the data structure may be important to the things that we do. Again, Giuliano's approach, I think, is more elegant, but Connor doing it by hand is fine too. I think it's very useful to see both options. Does anybody need help getting this to work? I see that there were still two people who said they wanted more time. If anybody has a question or who needs help, please speak up. Don't be shy about it.
No, one of the persons that answered that was me. I think it was because I understood something differently. I plotted not only the mean, but also like each single cell volume and surface. That's another question that I have. There is a simple way of like plotting every single cell volume, or usually we just do the average, the mean.
So here I'm doing exactly that. I'm doing the mean down here, but each cell I'm adding to the plot window V for volume. I have here cell.volume and plot window S cell.surface.
Okay. Yeah, I did that.
Yep.
Thank you.
Did anyone ever get any of the legends to work? I tried it on a desktop installation and on NanoHub, and I never could see any legends. Did anyone else have that issue?
The legends are working here when I do it now. Let me see. Does somebody else have the legends not working? So if I look at my output, I see Monte Carlo steps in the bottom and variables on the vertical.
So you were talking about
average volume of surface.
There's a special thing you need to set for it to set the legends. I can share my screen.
Oh, you mean actually labeling what blue and red mean?
Yeah, not
Okay.
not just like the X and Y labels, like the legend for the like data points.
Okay. Alex, go ahead if you want to screen share. I'm sorry, I misunderstood your question. I know you emailed me about it, and I thought I might be misunderstanding. And
You have
sure
to
enough,
add, I had to crawl through the code examples to find this. You need to pass this option to the new window creation.
Okay. Yeah, I tried and I couldn't find it, so I just tried setting legend equal to true, and that didn't work, so I just gave up.
Yeah, I tried that too.
Okay. So config
Really?
options dictionary. Okay.
Giuliano, can you make a note of that and make sure that we make our probably add a slide explaining that, and also make sure that that's called out in the manuals more clearly. These plots are using not matplotlib, but a different plotting library, and we'll find that there are a lot of options, some of which work and some of which don't, and so we're going to face that. And I have to say, I don't think I ever turned the legends on, and so probably a nice idea to do it, but I don't usually do it, and so I didn't think about that, and I didn't realize that that was what you were trying to do, and I apologize for that. Thank you for checking on it, Alex, and that's very helpful. Appreciate it.
Okay, yeah, I got it to work. Thank you.
Okay, anybody else having an issue? Because we're going to take this and continue with this, so it's important that everybody have this working, because everything going forward is dependent on it. If you're really having problems with it, there's a version of this called exercise 4.1.1 underscore 1.1 in that student materials folder. And so I want to make sure that everybody is okay before I move forward. Does anybody need more help or time? Again, don't be shy. Okay. And my screen share is all right. You can see the slides and the
Nothing sharing right now.
Nothing. Oh, because I unshared to. Thank you. I unshared because I was. There we go. Now is it okay?
Yep, we're good now.
Okay. All right. So again, my version of this was this naive version where I did what Connor did, simply iterated and subbed. So to do that, we had to change the target volumes. The defaults that we get from the wizard were setting the target volume and the target surface to 50, and so we had to make those to 125. Now what I'd like to do is go forward. I'd like to specify the volumes and the targets in Python instead of in XML. And so to do that, we still need to say that we have a volume and a surface, because nothing exists if we don't assert it. And so we still have to have the XML that says that we have a volume constraint and a surface constraint. But instead of giving the values here in volume energy parameters, we're going to do that in Python. And so what I want you to do is you can delete this line or you can comment it out, but you'll have something that looks maybe a little arbitrary. It'll be plugin name volume, end plugin, or if you want to use the shorthand, you could say plugin name volume backslash and a slash and close the tag that way. So delete the value specification here. And this will tell CompuCell that it needs to calculate the volume, but that the volume is going to be given in Python rather than here. In the case of the surface and the volume volume constraints, it's relatively unproblematic. I just managed to delete the line that I needed. It's relatively unproblematic that you delete those values. There are going to be a couple of places where you have to delete part of the specification, but not all of it. And occasionally it will be unclear what needs to be deleted. In particular, when we do chemotaxis, for example, it may not be so clear what has to be not specified. But in the case of volume and surface, all we do is delete that middle line. And so can I ask people to try that out? Is everybody able to do that? And if you save that and now we try to run it, we'll see that my cells shrink and disappear because there is no target volume and target surface anymore, because we haven't specified what those values should be. Okay. And now I get a division by 0 because I have a 0 volume and 0 surface. All right. Did everybody have that work for them? Anybody have a problem with that? Again, all I did was I went in and took the specification of the initial the volume constraint and the surface constraint, took out the values. And then I didn't ask you to do it, but I ran it and I saw that the cells lost their volume as time went on. Okay. To set the initial values inside of Python, we are going to use our usual iterator for cell and self.cell list, and we'll set the attributes of the cells: target volume, target surface, lambda surface. And so here we will go into the Python. We will go into our start function. And we can put in our iterator, visit all cells, as usual. And now for each cell, we are going to say cell dot target volume, capitalization matters. So my typing matters here, which is a bad thing generally. And here I said use 25 or 50. Why don't we do 125, which is what we were doing before, 125. Cell dot target surface equals 125. Cell dot lambda volume equals 2. Cell dot lambda surface equals 2. Okay, is that clear? So we're now iterating over all the cells, and we're setting each cell one at a time: target volume, lambda volume, target surface, lambda surface. So now we could have every cell have a different value. And here, if I wanted to, should be a new version, so I can do save CC3D project as, and I can call it lecture I call it, what's exercise 4_1_2. So let me make sure that that worked for everybody.
I had a quick question.
Please go ahead.
So I actually did this for one of the homework problems, so I didn't have to run a simulation twice. But I found on the online that there is like a function called like .get XML element that if you like assigned an ID to one of the XML, like the target volume XML tag, you could change it. Is there any benefit to doing that over this, or is it essentially doing the same thing? Because I don't know. Yeah, go ahead. Sorry.
Very good question. So if you want all cells of a given type to change together, then using the XML element is perfect. Because if I change the volume, target volume or target surface in the XML, I'm simultaneously changing it for all cells of that type.
Mm-hmm.
And so if I want to manipulate all of the cells together of a given type, then doing that XML manipulation is what I want.
Okay.
If I want to have something where eventually I'm going to have a tumor where one cell grows at one rate and one cell grows at another rate, then I want to be able to change the individual cells separately. And then I probably want to define things by cell type.
Okay.
And so in principle, there's a scoping issue, and CompuCell is not very orthogonal in the way it does scoping. So you can typically define something that's true for all cells. You could define something that's true for all cells of a given type, or something that's true for all cells, for individual cells, cell by cell.
Mm-hmm.
And if I were going to build this language specification again, I would build it in a little bit more consistent way so that you didn't define the by type and globals in XML and the by individuals in Python.
Mm-hmm.
And I would also probably allow you to do the initialization using the by type and then override it with individual values. And there are certain parameters you can override individually in Python, even if they were set in the XML, and some you can't. And I actually don't always know which it is, so I'd have to try it. Okay. Typically, we're going to do a lot more where we manipulate individual values. And that's going to mean we need more iterators. Because in your case, changing that one XML value will change all of the cells at the same time. Mm-hmm. Here we would have had to do an iterator to get those same effect. Mm-hmm. And so it's faster, computationally faster to do it the way you did it. And in a sense, it's more Pythonic to do it that way. But here we're doing the iteration by hand. So that's a good question. That ID label is fairly new. It's something that I had wanted for a long time, and that was resisted vehemently by TJ and Maciek as coders for a variety of philosophical and practical reasons. There was an old format of how you could change XML values. It was rather cumbersome. And the new one is a little bit better. One thing that we will have, which you'll see here, is that you'll notice now that if you run that simulation, it hasn't changed anything. I've got exactly what I had before. If it did change it, there'd be a problem. So then I'd have to change. One thing that I'm sorry, I lost my train of thought on that. We were talking about one thing that you'll lose when you do the Pythonic specification is that you won't have the steering option. So if I go into plug in volume, I can't change the volume on the fly during the simulation anymore, because this model editor only edits the XML specified parameters. Now, a little bit later in the class, at the moment, we can change these things by editing the code, so it's not a problem. We will find a little bit later on when we go to window. Let's see. I'm sorry, I'm in the wrong place. Okay, it's not there. It's slightly different in this version. If we go to CC3D Python, here we will find that there's something called a steering panel. And the steering panel will use basically an IPyWidget type interface to pop up a dialog box that will allow us to change Python variables and parameters on the fly. But in order to do that, we will have to explicitly declare which variables we want. They won't be made available automatically. And when you're doing your design for projects in NanoHub, since the user won't have access to editing the files when it's presented as an application by default, you'll use those dialog box, those steering panel boxes, as the way that the user can then do sliders or type values to change parameters. And so we'll come back to that. We'll come back to that in a few weeks. It's not something that we need typically when we're doing programs for ourselves, but if you're doing a packaged program, it's something that's quite useful. Okay. Any other questions about that? Those are good points. Okay. So the next thing I want to talk about is how we can store variables and information in CompuCell. And there are going to be two levels at which we do this. Again, it's a scoping issue to some extent. We are going to find that one of the things that CompuCell has that's quite convenient is that every cell comes prepackaged. I say nothing is for free. A few things are for free. It used to not be for free, and it was a pain. Every cell comes with a dictionary. And is there anybody in the room who has not used a Python dictionary?
I mean, beyond the last meeting we had, I would fall into that bucket.
Okay, so dictionaries take a little bit of getting used to, perhaps, but they're a very powerful concept. So we've had the idea of a list, in this case, a list of pointers that tell us what the cells are. A dictionary is a generalization of a list where instead of having an index that says the first element in the list, the second element in the list, the third element in the list, the elements in the list are referenced by a name, in this case a string, otherwise known as a key. And so in this case, I could have cell dictionary square bracket, because it's behaving like a list, attribute name, equals, and then a value. And that may seem abstract. I could say cell dictionary, quote, a name, close quote, equals hello. So I can give one element of my dictionary a string attribute. I could give another one value 10.6, a float. I can have one element be a list. 10, 11, 13, 15. And so I can now attach to my cell a set of user-defined attributes, which could be very simple, like alive or dead, a Boolean, or something very complex, like another dictionary. One thing you do not want to try to put into a dictionary of a cell is a handle. So one thing you will never do, and I've done it, so I can promise you it won't work, is to do something like cell.dict semicolon square bracket quote cell square bracket equals cell. Don't try to load a reference pointer object handle into a dictionary. Otherwise, you can do almost anything in a dictionary. You can add things to lists. You can search lists. You can delete things. I said lists. You can add things to lists. You can add things to dictionaries. You can append things. You can sort them. You can remove things. And you need to look in the standard Python tutorial or manual to see all of the different operators that you have. You can ask, what's the biggest element in a list? What's the smallest element in a list? What's the average value of a list? And so on. The one thing you have to worry about in an object-oriented language like Python is that list elements and dictionary elements don't have to have the same type. And so you can create a list where the first element of the list is a number, the second element of the list is a string, the third element of a list is something else. And of course, if you then try to operate on that with something that expects it all to be numbers or all strings, you're going to have a problem. That's really a discussion of Python rather than of CompuCell. The key thing here is that we can create a cell attribute, and while it may be cumbersome because it's a long definition, this kind of attribute is available to us to use as a variable. And there is a command, cell attributes access modify dictionary attribute, if you forget how to do that. Something that may seem less obvious, but that we will have available to us also, is that there is going to be essentially a global dictionary that's available for putting things that we want to use for the whole simulation. You could ask, why not just have a variable X defined in my Steppable? And the answer for that is going to be, and I'll come back to it, I have a different slide for it, that variables that are defined inside of one class are not accessible to other classes. And so if I create a value size of simulation that lives in a Steppable, one Steppable, that variable is not automatically available in another Steppable. If we only have one Steppable, it doesn't matter, but if we have multiple Steppables, it's an issue. That's sort of technical, but we will run into that when we do cell division. So now that we have this function, I want to plot the pressure and the surface tension when cells grow or shrink. And so I want to remind you that we defined the pressure as the derivative of the volume constraint, which is -2 times lambda volume, V target minus V. And the surface tension or membrane tension is -2 lambda surface S target minus S. And for a long time, we had to calculate those things by hand. And relatively recently, they were added, so cell dot pressure and cell dot surface tension exist. So we don't have to keep doing that calculation. The one thing that you're going to find is that the sign of this is the opposite direction from what you would expect. And so if the actual volume of the cell is smaller than its target, we're going to get the opposite sign of what we might expect. The pressure is defined inward rather than outward. And so you'll see that you'll have to get used to it. Maybe it will make sense to you to have a direction that it is. And so what I'd like people to do now is to create in the simulation you have an additional plot window, or maybe two, one that plots the pressure and one that plots the surface tension. And let's follow, just by templating, exactly what we've done before, a scatter plot of the pressures of the cells one at a time and the mean. And so to do that, we can make our lives easier. You can simply grab all of the plot window stuff that you had here, cut and paste it. Remember that when you cut and paste it, you're going to have to change the handle on plot win to give it a new name. And you're going to have to change the title of the plot to something like surface tension and pressure. Can't keep it there. And then the same thing can be done down here, where we can create, add the data points for the surface and volume, so the pressure and surface tension, and then either using Giuliano's more elegant method or by hand, calculate the means for the pressure and surface tension. Okay. And doing that initially is not going to be very exciting. Like, it'll be pretty boring. But what you can do after that is the last thing to do is we want to increase the target volume. The whole point about defining the target volume by cell is now we can change it by cell. So what I'd like you to do is inside your cell loop, I want you to increase the target volume. Here, I'd like you to increase the target volume by 5 every 10 Monte Carlo steps. And there are a lot of different ways you can check if you're every 10th Monte Carlo step. It's not the most beautiful syntax in the world, but I find that if not MCS %10 colon is a good way of doing it. You could define a steppable that's called every 10 Monte Carlo steps, which would probably be more elegant. But percent is integer division, and it gives you the remainder. And so MCS %10 is MCS modulo 10. And that will be 0 if MCS is divisible by 10. It'll be non-zero otherwise. And so not of that will be 1 when MCS is a multiple of 10 and 0 otherwise. If you have a different way that you like to count to 10 by tens, that's okay. Okay? And when you've done that, I'd like you to tell me what happens to the average in individual volumes and surfaces and the average in individual pressures and surface tensions. So is that a clear assignment? And again, if these little exercises are either going too fast or too slow, you can either speak up, or if you feel that that could be controversial, you can do a text message to me in the chat saying, hey guy, it's too fast, or hey guy, it's too slow. So you don't have to let anybody know who said it. Again, if you get stuck, these are available, these simulations are available for download from the student materials.
So I think my code should work, but I was trying to initialize counters just because I like to have those so I can actually print trials usually when I code. And I put it in the start function, but maybe that was the wrong place to put it. It did not like that.
So what are you trying to do? You want to screen share it, and we can look at
Sure.
it together?
Yeah, it's more of a brain glitch than anything. I don't think it should actually be an issue. I was just trying to figure out where to put these. I had a variable called MC counter and pump that I was just trying to initialize at the start of the run. Because down here, the way I had it set is that I have a counter that tells me when it's time to increase the volume.
Okay, so I strongly urge you not to do that.
Okay.
Because the Monte Carlo steps is a global that's available everywhere.
Okay.
If you want the variable to be available everywhere, you need to define it outside of the start and step functions.
Okay.
So you'd have to go up to the top at the beginning of the of the code, because functions are functions, and so things that are defined inside of a function are local to the function. So this is something that you have to remember, that functions are in fact functions. So if you want things to be available outside of them, you have to put them up at the top of your code outside of the function.
Or declare it as a self variable, so it belongs to the whole class.
No. If you define it as self dot, it'll be available for the whole class. This is again Pythonic, and if you really care, you can make things globals. Although the use of globals in Python is a little bit fraught. They don't behave quite as the way you might expect. The other thing to do is, and we haven't talked about it yet, is you could create a dictionary entry for the whole system, and in that case, that dictionary entry is available globally.
Okay.
But I wouldn't recommend using a counter like that, just because you're duplicating a functionality that's already there, because the Monte Carlo steps are there. Now, it's true that sometimes you want to do something where you want to know how long it's been since a cell divided. And in that case, you'd probably want to store the time of last division as an attribute of the cell. So in that case, that kind of counter would be used. Good point. Any other comments or questions about this?
What is the tension graph supposed to show?
So the pressure is the difference between the target volume and the actual volume. The tension is the difference between the target surface area and the actual surface area.
All right, because all my graphs work, but the tension one doesn't show anything when I run it.
Okay, let's see. Take a look together.
So I have all my other graphs run, but the tension one doesn't show up.
Okay, let's see your code. So you created AT so you're storing the tension in the data series is supposed to be TNS. Is that right?
Yeah, and then AT was going to be the average.
You're putting those lines on the wrong plot. That's self.sur, not self.tension.
Oh, that's what it is.
So yes, that was what I was going to say. If you add the series to something that doesn't exist, it doesn't throw an error. It doesn't display it. Okay, so let's see what happens there. Did it crash or did you hit run?
I hit run, but I have like the little death circle. So
Okay. So that means there was an error. Let's look at your code again and see if we can figure it out. So let's look at the where you created the plots. You create a new plot window. Slow down, please. So you create a new plot window. Pressure, you create a new plot window. Tension. Okay. In pressure, you put pressure and AP. Okay, that looks all right, I think. Juliana, if you see anything, speak up. And let's go down. For the second one, you create tension. And you add tense and AT to that. Okay, that looks okay. And now when you add the data points, tense, pressure, okay. And then you add mean of P, mean of T. Giuliano, do you see a bug in that? That looks all right to me. Why don't you start restarting it and see if it'll work. Again, okay. Oh, I should have said we should look at the console before.
Oh, sorry.
Did anybody else have any problems with that? Okay, it opens.
I think it works now.
Okay. All right.
All right. Thank you.
Okay. All right. We've overrun our normal break time, but I want to get through one more thing before we take a short break. Anybody else need help with this? Again, the code is available if you need it in the student materials folder. So again, here's the way I did it. And I think we've seen this repeatedly, so I'm not going to go through it. The next thing I'd like to do, which I talked about, but we actually haven't done yet, is only one line, which is create, when you go through your code, I would like you to increase the target volume by 5 every 10 Monte Carlo steps. So why don't we do that and play with that? So to do that, I'll do it by hand here. When we go through self.cell list here, I didn't do the every 10 yet, so I'll put that in myself. If not, MCS %10 colon. I'm not very good at typing and talking at the same time. And now I'm going to increment my cell target volume. Okay, so let's do that. And this will probably not be very pretty. So I would recommend doing what I originally said in the code, which in the slide, which is not what we did here, which is I'd start with my target surface volume being 25. And my target surface being 25. Actually, I said 50, so let's make it 50. You can play with that a little bit. Okay. You'll notice here I started with the cells being much bigger than their target, so they're shrinking. And then little bit by little bit, you'll see that the target volume is coming up again. You'll see these glitches every 10 Monte Carlo steps as I adjust the target volume. And you'll see that the average volume will gradually go up, and the average surface will go up. And this may be clearer if I make the cell field bigger. So they're my cells. Were people able to get that to run for themselves? Okay. Can people explain to me what's happening with the pressure and the surface tension?
They aren't really changing relative to each other because we're increasing the volume and surface area at the same time, at the same rate.
Okay, so you're increasing the volume and surface area at the same rate. I didn't increase the surface.
Oh, okay.
And so what's happening here is that the mismatch for the surface is getting worse and worse, because I've tried to squeeze more and more volume into the same surface. So I'm blowing air into a balloon, and or into a beach ball, and it's getting overinflated. So you'll see that these cells are getting more and more rigid and square because I keep trying to pump force volume into them without adjusting the surface. Now, what you've suggested is the next thing that we should do, which is that we should probably have the surface area increase as well as the volume. And so if I do that, let's increase the target surface at the same time. Now I shouldn't see this divergence between pressure and surface. Everything's shrinking initially because I set my targets to be 25 instead of 125. Everything will shrink, and then they'll reach an equilibrium, and then they'll start growing again. And now you should see the things behave better. And you don't see that divergence between pressure and surface that we had before. Now you see volume and surface are both increasing stepwise the way you would expect. What will happen pretty soon is that the cells in the middle of this cluster will be pushing on each other, and so they're going to be compressed compared to everything else. And so what you could see happen is that the deviation of volume and surface could get bigger as I go forward, where the cells in the middle aren't able to expand. Having said that, it may not do it. Maybe I'm wrong about that. Now you see now the pressure and surface tension are more or less constant. The volume and surface are both growing. The pressures are relatively constant. Was everybody able to get that to work? Yes? No? Does anybody need help with that? Why don't we take a 5-minute break, come back at 7:11, and I guess if anybody has a question, I can task Giuliano with answering the question during the break. How's that? Poor Giuliano doesn't get a break. Okay. People could play with this. For example, here we've increased the volume linearly, but you could do cell.target volume times equals 1.2 and have an exponential increase in volume. Or you could play with having the volume and the surface area change in different ways and see what happens to the pressure and the surface tension and the actual and target volumes as time goes. So in a sense, this is a very flexible simulation because we're monitoring a lot. Okay, so I hope that was making sense for people. And there are a couple of more plotting things I want to go over today. Maybe we won't get through all of the. We should look at the homework together to see before we break at the end, to see which problems make sense given what we cover. It's not a huge deal one way or the other, but I want to make sure that people are comfortable with the things we cover. Okay, let's get back here. Is everybody back? I could say, is anybody not back? If you're not back, say something. Okay, so the next thing I'd like to do is talk about a very powerful function in CompuCell, which is called a tracking field. And this is, I think, probably the single nicest feature that CompuCell has. So it's pretty common, both in experimental biology and in modeling, that you want to know something about the state of a cell. You might want to know how much ATP there is in the cell, or you might, in this case, want to know how big the cell is, or what the pressure of the cell is. And CompuCell has a single function that allows you to display any scalar or any vector quantity associated with cells on a cell-by-cell basis. And that's all embodied in a single line of code. And that's called a tracking field. And the tracking field, as usual, is available in the Twinit menus. So I'll come to the Twinit menus. And unusually, the tracking field has to go into the init function. We're used to putting things in the start of the step function, but the tracking field goes in the init. And there's a little bit of a mine here, which is that you'll see def underscore init, and then you'll see an empty line, and then you'll see steppable base.py.init. And it has to go after the steppable base.py dot. And there probably should be a default boilerplate that says put your code after this line, or at least get rid of that empty line 7, so that nobody's tempted to paste things in above it. So here we're going to go into our init. We will go into CC3D Python, and we will go down to extra fields, automatic tracking, track scalar cell attribute. Okay. And we will paste that in. And we will see that it will paste in this line, and we will give the field name a name, and an attribute name an attribute name. And in this case, we're going to be plotting the pressure. And so we will call the field name pressure. And the attribute name could be something that we like. Let's give it also the name pressure. Then, somewhere in our code, we will create a cell dictionary entry that corresponds to the attribute name we used creating the field. And the one thing that we might find slightly inconvenient here is that in the case of the pressure is an intrinsic attribute of the cell, and we're going to have to copy it into a dictionary entry called pressure. If we picked something that was not an intrinsic property of the cell, it wouldn't seem strange to do this, but here we have to copy something back into the cell. And so here, when we do our function iterator, in our step function, we will, in addition to where we are measuring the pressure, we will say cell.dict, and now I have to remember what I typed, pressure equals cell.pressure. And I could have added cell volume. I could have added something derived, volume plus pressure, volume squared, any attribute that I like. The only thing that's critical is that the word pressure here must match my declaration up here. Again, what I have in the PowerPoint slide gives you the syntax. Whatever is given the attribute name here, you have to create a cell dictionary entry with the same attribute name. So why doesn't everybody try that? So you're adding 2 lines. In the init, you're adding track cell level scalar attribute. Remember that comes from CC3D Python. Extra fields, automatic tracking, track scalar attribute. And in my step function, I am copying the cell pressure into a dictionary entry called pressure. And I could create, again, as many of these as I like. I could create a tracking field called tension for the surface tension. I could create a tracking field called volume, which had the cell volume in it. Actually, the volume is sort of an interesting one, so that would be one that's not a bad one to do. And let's see what happens now when I run that. Well, it looks like nothing happened. So, why does it look like nothing happened? Let me hit pause in the execution of my simulation. If I now go into my graphics window and I go where I have my cell field name and pull down here, I'll notice besides the cell field, I have pressure offered as an option. And it doesn't work very well in 3D, so I'm going to go back to a 2D image. And I now have a pressure in every cell. And I probably need to set up that field under settings so that it auto scales. So why don't people spend a minute and play with that? And I realize that the key and the access label are screwy. I'm not quite sure why the color bar isn't right. There's a scaling problem with the labels. So that's actually a pretty powerful function to have.
Because we're doing to all cells, is also counting the medium as a cell. Wouldn't he? There's a way of doing it like just for the cells by type or something.
Yeah, you could exclude the medium if you wanted to, yeah, and have it not display. Here, the medium will have a value of 0. And so, so in a sense, that's not unhelpful because red, then you know red is 0. But yes, you could exclude the medium if you want.
Yeah, because on mine is changing over time. Like it's becoming other colors over time.
That's probably
So becoming other colors, I don't know, but medium is not technically a cell. So
Yeah.
CompuCell in that case doesn't know what to do, so it puts 0 there.
Mhm.
instead of just leaving
Yeah.
it black. So the loop over all cells then does not loop
Doesn't.
over medium.
Oh, I'm sorry. Yeah. My
Oh, okay. I thought it was considered like a cell with ID 0, but okay.
No.
So that's actually an issue, that you can't attach certain things to medium, because medium is a null pointer. And I should have said that. If you want to attach things to medium, you have to create a medium 2 cell. And that has a particular disadvantage when you parallelize CompuCell, which I don't want to get into. But
Okay, thank you.
thank you for correcting that, Juliana. Did people get this to work? Did anybody have it not work? Maybe it's a better question. Looks like everybody says it worked. So why don't you try one other tracking field? Why don't you pick something that you want to track? Cell volume, cell surface area, cell pressure, surface tension, and plot, add that too. I don't have that on the slide, but just to check whether things make sense, I'm going to do cell volume. This one is particularly useful when you're doing subcellular networks, because the variables in a network model of gene regulation or signaling or metabolism are dictionary entries. And so in that case, you have the analogy of a immunocytochemistry or immunofluorescence experiment, where the cell is automatically labeled by the concentration of the chemical. And in that case, since there's already a dictionary entry for the variable, you don't even have to add the second line. It takes one line to display the level of ATP in the cell, for example. And so from that point of view, it's pretty powerful. I've actually never used the vector one. I don't know how it works. So we could try that at some point.
It's not that pretty.
I wouldn't expect it to be pretty, but it's there.
Yep.
So here I've added the volume of the cell. And let's see whether that works. Again, you may have to go under settings and make sure that the field has a float for the range, assuming that that's what you want. So that's how to use a tracking field. As I say, I find that one of the most powerful convenience functions in CompuCell. So we've done this. The next thing about plots, and I guess today is going to be heavy on plots, but at least once you've done this, you know how to do a lot of plots, is histograms. Here we have sort of a poor man's histogram because we have a point for each cell. But we might want to bin the volumes or the pressures or something else. And so to do a histogram, of course, is a plot of the frequency of occurrence of some value, some scalar value. And we're going to do a histogram of the pressure. So we're now going to have the pressure shown by individual points, the mean of the pressure, a tracking field for the pressure, and we're going to do a histogram of the pressure. So we're going to have the pressure displayed in a whole variety of ways. And there probably are some other ways we could plot it too, but this will give us pretty much all the things we would like to do in terms of reporting for pressure. Histograms are going to work very much the way regular plot windows work. We're going to go into the start function. We're going to add a setup for the histogram. Then we're going to go into the step function. And in the case of the step function, a histogram is taken from a list. And so we will pass to the histogram plot a list of the things that we want histogrammed, and it'll do it automatically. And again, in principle, we can restrict a variety of things on the histogram, but the defaults are pretty good. So to add a histogram, we are going to first go into the start function, find a spot where we don't have code after, for example, our other plot windows. And so we're going to find that very often when we're doing CompuCell, that setting up the plots is the most lines of code. We're going to have a lot of lines that set up the plots. The actual implementation of the simulation is quite short, but the plots take up a lot of code. On the other hand, they are going to have a structure that's pretty simple, which is going to be creating the plot window, adding the data series. You're going to see the same thing repeated again and again for all the plots. And so it's long, but it's not intrinsically complicated. You go to CC3D Python, scientific plots histogram, add histogram plot. So follow along and do that. And it's going to put in a bunch of stuff. And what we're going to have is a self.plot window. Again, we're going to have to change that handle because it's been used already. And it's going to give us add new plot window. And it's going to be histogram this time. And it's going to give us an x-axis, a y-axis. And then we're going to, in that plot window, add a histogram plot. Now, instead of a regular data series. And we'll give it a name and a color and an opacity, because when we want to overlay histograms, we have them, basically these are bar charts, and so we have to be able to determine whether we can see through them or not. For the moment, we're only going to need one, so we're going to get rid of the extra ones. So we're going to paste in the histogram command. We have to rename plot window to something unique. We're going to have as our x-axis pressure. Our y-axis is the frequency of cells with that pressure. And then we only are going to want one histogram, so we'll get rid of the two extras. So is it clear what we need to do? I've got it here. That may be small to see depending on your display. So I'm going to rename plot window to histogram window. Hist. The histogram is going to be of pressures here. The x-axis will be pressure. The names of the x-axis and y-axis don't matter in terms of code execution. The title has to be unique, as before. The y-axis title will be frequency. I'm going to get rid of these two lines that I don't need. Histogram 3 and 4 I'm not going to need. 2 and 3 I'm not going to need. I need to remember I changed plot win to hist win, so I have to change my handle here. Easy to get that one wrong. Okay, I don't need to change anything else there. And so does everybody follow that one all right? And now the step function is going to be a little bit more involved. Although the people who were already assembling lists of the pressures now have done the right thing, because you'll need it exactly the way you did it. Okay, everybody got that? I can come back to it if it's a problem. And so now we have to create a list which contains the things that we're going to histogram. So in our step function, I'll create something called pressure list. And I'll create an empty list. When I measure the pressure here, I'm going to use an append. Cell.pressure. And so now that's going to create a list. And now I wouldn't have to do the adding. Now instead, I wouldn't have to do I could get rid of this mpree equals 0, because now I could use that numpy function of mean if I wanted to. And the last thing that I'm going to do is I'm going to add my histogram. So I'm going to go down here to the end, CC3D Python, scientific plots histogram, add histogram. Again, it's going to put in 3 of them, so I'll get rid of the other 2, which I don't need. And you can follow it here on the slide, probably better than watching me type. I need to remember that I called this histogram window instead of plot window. Plot name was hist1, so that doesn't change. And now value array is the name of that list that I created, pressure list. And number of bins is going to tell me how many bins I'm going to put in my histogram. So is that okay for people? It's all here on the screen. If people need to go back and see the example before, I can do that. Stop. And we'll see if I have a typo. See if it works for you. And now I have a histogram. So this is the pressure in each cell, binned by 10 bins. And it's a live histogram, so it's continually updating. Did people get that to work? I see 5 people said yes. Does that mean everybody got it? And again, that transparency is because I might want to show a histogram of the pressures and the surface tensions on the same plot. And because it's a bar chart, if it's completely opaque, I won't be able to see the one behind it. And so you can use the color, the transparency to adjust the visualization. So this is going to be now a pretty powerful set of display tools that you can use. And you'll see that as time goes on, the pressures will change. After a while now, my pressures are basically what I see here. My spots are basically giving me the width of this distribution, and my pressure histogram of pressures would correspond to this histogram of pressures here with my spots. I can see a mean pressure of minus 110 and the width of it. If I went in and steered the lambda volume, I made lambda volume bigger, I would change the width. Something else that you might want, I'm often asking you for screenshots. You could certainly use a snipping tool to take a screenshot. You can save movies using the visualization, sorry, not the visualization, the settings bar, and we can come back to that. But something else you could do here is if you right-click over a plot, you'll see View All X-axis settings, manual versus auto, Y-axis settings, manual versus auto. Some of these work, some don't. So I won't promise that they all work. Plot options, some of these don't work, I know, like the transforms don't work. So they should really be grayed out. But the one that you want is export. And if I hit export, it will pop up a dialog box and it'll give me some options. Image, scalable vector graphics, matplotlib window, and CSV from plot data. If I hit have image and I hit copy, well, it crashed in NanoHub. Does it crash for you in NanoHub, Juliana?
I was doing it local. But I've done that in NanoHub before and it worked.
I gotta terminate. So that may only work. That may only work in your local machine.
At least during last year's workshop, it worked on NanoHub.
Let's see. Does anybody else have experience of it working or not working? I know some people are running locally and some people are running online. So let's see what happens. Let me pause it just to see if that helps. Export. And I'm going to do a copy and it crashes it.
Locally for me, copy was not working, but instead of copy, if I hit export, then it opens a folder that I can name the file, and then it worked.
Okay. So the second one that I wanted to do, so Giuliano, please take notes about these glitches, because these are things that we need to fix. So locally on my Windows machine, I have no problem with copy. And it will put a copy of the image into my clipboard, which I can then paste. So instead of having to do a screenshot, I can do that way. Anybody else want to report their experience? But what should work is if I go to export, and now I select export, it will give me the option of saving it. So that will work. So what seems to be an issue is that NanoHub doesn't give me access to, well, remember, we can't cut and paste well in NanoHub anyway. So there's some privilege issue with the clipboard in NanoHub.
Yeah, copying on a virtual machine is always a problem. Yeah.
But I can save the image for further use. The other thing I could do is I could go to CSV from plot data, and I can hit export, and I can save that, and that will give me the data set. That may not be so useful for the histogram, but here, let me run it for a while. For pressure and surface tension, this is a lot of data points. And I could certainly, and I'll show you how to save data from an executing simulation, opening a file and saving it. But it's a little bit elaborate to do that. And if you want to try things out to decide whether it's worth saving it from inside the simulation, you might want to have access to this data set to play with it offline. And so here, for pressure and surface tension, I can go to export, select comma separated value, export, and then I can save. Remember, I have to save to my home directory, not to the root directory where I don't have write privileges on NanoHub. I can save this entire data set of all those data points and then download it for further processing. Or if I open up something like a Jupyter Notebook window, I can pull that data set into Jupyter Notebooks and process it on NanoHub. And so that's a pretty powerful thing. And so when you're preparing your homeworks, if you've been screenshotting rather than saving images, this is pretty convenient, especially if you want to do post-processing. And so I wanted to show you how to do that. And again, I apologize for the fact that the save to clipboard doesn't seem to work, but you can save the image. The other thing that we might want to think about here, again, is under configuration, we can go to output, and you can do save images. So you can save the image every n Monte Carlo steps. Here you have to be a little bit careful about where you put it on NanoHub. You have to make sure that it's pointing to a directory where you have write privileges. But here you do, because it's under my name, so I have write privileges. And I have a choice. I can save the image. I can save visualization snapshots. And they're going to give me slightly different information. You can play with that. Saving movies on NanoHub is not as convenient because then you have to zip them to download them, because it's going to save a whole series of individual images. Each screenshot will be saved as a separate image. And then locally, you're going to have to use Microsoft Windows has a tool for turning a stack of GIFs or a stack of JPEGs into an animation, into a movie. It's a different tool on a Mac.
Just out of curiosity, so
Sure.
This image export, are you allowed to choose what kind of format these individual images export as, or is it by default just JPEG?
The save here, actually, what is the format it saves it in? Is it a GIF? TIFF? It's a TIFF, isn't it?
Yeah, TIFF makes sense. I think GIFs are like just
For the
for
animation.
moving images. Yeah.
It'll be a TIFF here. If I use the right click, let's see what it gives me as an option. It looks like I can select export, and I have a choice. I have a lot of choices.
Oh, okay, that's perfect. All right. Thank you. Just wanted to ask about that for like
Yeah.
presentation purposes and stuff.
PNG, TIFF, JPEG. I've never heard of some of these formats.
Yeah.
ISO.
Does that
Bitmap.
extend to the cell field capture? Because when I use it, it always defaults to PNG, and I don't know, like on the, not with, if you just like right click and export, but if you like actually use the visualization to save
Yeah.
image on nth Monte Carlo step, I don't see anywhere where you could specify the type.
Okay, so there is no right click on the cell field. On the fields, this is the good and the bad about the way scientific plots are implemented. As I mentioned, scientific plots are done using a utility like matplotlib, although not matplotlib. And so these saving functions are intrinsic to that utility. They're not things that we wrote.
Mm-hmm.
The rendering of the cell field is rather complicated because in the simulation proper, what you have is an array of voxels, each one of which is numbered. 111, 000, 111, 222, 333, 444. And so to do this rendering, there's a rather complicated pile of C++ that says, where are the cell boundaries? What color should each domain be? Do I draw the edges? Do I shade it? And so on. And so all of this rendering is done essentially by hand by CompuCell. There isn't any standard package that will do that rendering for us. And so the saving here is done through this options here. And we don't have, at this point, the option of changing the output format. Probably we should have a save as TIFF or save as GIF option, but we don't. So the answer is you're stuck with the PNGs for the outputs for the visualizations. If somebody wanted to write some exporters, that would be great, but we don't have. Okay. Any other questions about plotting? These are all good questions to ask, and I hope Giuliano is taking notes so that we can put these requests on the pull request list, especially when you find a bug. It's important that we know it. All right. So I think we're almost out of time here. One thing that I wanted to come back to just from the homework, and this is again really from 2 weeks ago, so it's the homework I just graded, is that the first thing that I did in the homework, and I think we went over it a little bit last week, was that I asked people to calculate by hand the surface area of 2 images. I asked you to calculate the surface energy of some images. You had to calculate first the volume energy and then the surface energy. And everybody had the volume energy just fine. But some people had a little bit, because counting the voxels is easy. Counting the surface is a little bit more problematic. And the reason that the surface is a little bit more problematic is that when you do the surface plugin, one of the attributes of the surface plugin is the range. And let us look at this picture. Here I have a single voxel, so the volume is 1. I'm assuming I'm in 2D. The volume is 1. The surface for nearest neighbor is 1, 2, 3, 4, and I can figure that out, that when if I have nearest neighbors, it's simply the number of edges or faces that I have. So my volume is 1 and my surface is 4. If I now have 4 by 4 voxels, my volume is 4, my surface is 1, 2, 3, 4, 5, 6, 7, 8. If I now have a volume of 21, here it's a 5 by 5 square with the corners chopped out. I count around the edge 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So my perimeter is 20, my surface is 20. If I added these 4 corners, what would the volume be? Not a trick question.
25.
25. What would the surface be?
21 or 20.
20. So I can adding 4 pixels to the corners changes my volume by 4, but doesn't change the surface area at all. And so this is one of these things that's a little bit counterintuitive about the way volume and surface work in a lattice. There's an approximate relation which we talked to in 2D. The minimum surface for nearest neighbor for a given volume is essentially the surface of a square. And so if I have a volume of V, and I think about arranging that in a square, the minimal surface that I can have enclosing it is 4 times the square root of V. So here I have 1, square root of 1 is 1, times 4 is 4. Here I have 4, the square root of 4 is 2, times 4 is 8. Here, imagine I filled in my corners. I had 5 by 5. So my volume is 25. The square root is 5. 4 times 5 is 20. So the perimeter is 20. If the surface area is smaller than 4 square root of V, there's no way I can satisfy the surface constraint and the volume constraint simultaneously. My target surface is less than 4 square root of V. I can't satisfy them both. There is no configuration which has that small a surface. Similarly, if I imagine dispersing my voxels, and I should add that to this slide, if I put my voxels all independently and my surface were bigger than 4 times V, even if I dispersed my volume into individual voxels, the target surface would be bigger than I could achieve. And so if S target is less than 4 square root of VT, I'm always squeezing my volume from the surface constraint. If S is bigger than 4 VT, no square root, I'm always expanding from the volume. If S target is between 4 square root of VT and 4 VT, then it's possible to satisfy both at the same time. And then the lambda volume, lambda surface will matter. And when I gave you an assignment, when I said if V is large compared to S, V target is large compared to S target, that's what I was talking about. Large means bigger than or less than 4 VT, bigger than or less than 4 square root of VT. And in that middle region, those lambda volumes make a big difference. On a 3D square lattice, it's a little bit more complicated. Now I have a cube, and so it's 6 times VT to the 2/3. For the lower bound, it would be 6 VT for the volumes being separated. Does that make sense to people? I know we're at the end of the class, so I don't want to go too far. Okay, and I don't want, this is what happens when you play with that. I don't want to go too far with that. The one thing that I wanted to say beyond that, and I don't have the slide, it's a little bit further down, is when you go to second neighbor, it's a little more complicated to count. If I take this cell and I ask how many second neighbors of medium it has, this one cell will count 1, 2, 3, 4, 5. So that one cell corner will contribute 5. This will be 5, 5, 5. The surface area of this 4 voxel volume with second neighbor interactions is 20. And so in that second homework problem where I asked you to say what happens if you go to second neighbor interactions for the surface, the counting gets a little bit more complicated because you have to count the second neighbors for each one, and the voxels in the neighborhoods are get counted more than once. This one that's labeled 2 is a neighbor of this voxel and this voxel. This voxel neighbor labeled 4 is a neighbor of this voxel and this voxel. And so counting the surface for longer interaction ranges is a little bit more complicated. And that's why I gave that homework problem specifically. The other thing is that surface is any boundary. If I make a hole in the middle of a cell, it's still surface. And so any mismatch between two lattice sites counts as surface, wherever it is. And so some people had no problem with that first homework problem. Some people found it a little bit confusing. And it's okay to be confused, but I want to make sure that I explain that as clearly as possible for that. Any questions about that? The last thing I want to do, and I know we're over time, is come to the homework assignment. Did the homework show now? Is the homework assignment?
Yeah, it's showing.
Let's look at the problems just quickly at the end here and see what we can do. I said adjusting surface constraints in growing cells. So this is doable because we know how to measure the pressure, we know how to change the surface, and so that problem is going to work. The next problem, hydrostatic pressure in the presence of cell growth and division. We haven't really discussed how to do cell division yet. And I think all of the remaining ones are also, that second problem involves cell division. I think I'd like to ask you to try to do cell division as a challenge exercise. See if you could figure out how to do it from the manuals and the examples online. And if you can, do the problem, and you'll be then the lecture next week will be maybe too simple. If you can't, then we'll cover it next week. Cell sorting and cell growth. This has also got cell division in it. So that problem has cell division in it. So fundamentally, I would ask you to try to do the cell division on your own. If you could figure out how to do cell division, and there are good examples, you can do the 3 problems. We covered it. If you can't figure out how to get cell division to work, do the first problem and focus on your project. Is that okay for next week then? But I do want to try to make sure that people aren't frustrated by the homework. On the other hand, I think it's useful to try it, at least give it a shot. And if it's not successful, you can ask questions and we'll cover it. Okay. Any questions before we break? We're over time, so I don't want to take more time than necessary.
It doesn't have to be right now, but I would appreciate at some point if we could go over the getting contact energy from the neighborhood. from the previous homework assignment, just because that was the one thing that kind of tripped me up. I think that my algorithm is roughly right, but I'm probably calling something wrong. But
Right. So I apologize. That was the next topic in this slide deck, and we didn't get to it tonight. But next week we will do it. So iterating over neighbors, iterating over cell neighbors is a little bit confusing because the neighbor iterator, and I guess I can show that. Let me just show that quickly here. I'll just paste the code in here in a random spot. CC3D Python visit. Cell neighbors. And it returns for neighbor, comma, common surface area in self.get cell neighbor data list of cell. Cell data list of cell returns something you would expect, which is the list of neighbors, but it also returns the area, the contact area with each neighbor. And so the one problem with that is if you'd said something like neighbor equals self.get neighbor list, it's not going to return the kind of object you'd expect. And so neighbor actually here contains a list, neighbor ID, and the common surface area for each one. And so when you do this, this iterator will go over everything, and it'll give you both the name, the identity of the neighbor, the type of the neighbor, and the common area with the neighbor.
So one thing that I'm confused about is how, like in the second problem, when we have to calculate like the contact surface area between like light, dark, and all those,
Yeah.
you know, permutations of it. How I don't know how you can discern, you know, light versus light dark and that stuff. Do you have to look at like the cell dot type and then all that, just like a bunch of if else
Yeah.
stuff?
So you'll have it. Exactly. I mean,
Okay.
you can be fancy, and you could have an array. You could create an array where you're indexing the array by cell dot type, comma neighbor dot type.
Mm-hmm.
But yes, typically you'll have an if statement where you'll have cell, because you've got cell and you've got neighbor, and you have neighbor cell dot type and neighbor dot type.
Mm-hmm.
And you'll have an if statement, if cell dot type equals self dot dark and neighbor dot type equals self dot light, then blah.
Okay. I think I found there is one where you could group neighbors by type. So I think I just like found all the cells of type light and then just calculated the area of it with itself and dark. And okay. I just wanted to make sure that I was doing it right.
The one thing that you have to watch out for a little bit is that if you iterate over all cells, contact area between cells and other cells will be counted twice, because cell 1 will be neighboring cell 2 and cell 2 will be neighboring cell 1.
Okay.
So the boundary between cell A and cell B will be counted 2 times.
Oh, okay.
However, contact with medium is only counted once because the iterator doesn't count medium. When you iterate for cell, it skips the medium. Medium will be counted when you do the neighbors. It knows about neighborhood with medium, but it won't be counted the other direction. You not get the source for medium. And so when you add medium contact, medium contact, you'll get half as big a number as you do for cell-cell contact if you do it naively. And so that's a minor annoyance because again, medium isn't included in that 4-cell self.cell underscore list.
Okay.
So it's not counted a second time. And so the cell contacts are not problematic that way, but cell medium contact is scaled differently.
Mm-hmm.
Other questions about that? And I will cover that next week, but those are great questions. Okay, well, I appreciate everybody sticking around. We started a few minutes late, but that doesn't mean running till 8:10 is great. I can smell food in my background. I hope you have food in your houses. I do appreciate your patience for this funny meeting time. And I appreciate your willingness to stick around. And I do enjoy, I hope today was a very engaged class. There were a lot of questions. And I realize that on the one hand, that means that we don't get through as much material perhaps. And maybe not every question is of interest to every person. But I hope that we can go I hope that most of the questions, in my experience, if one person is having an issue with them, everyone does, or almost everybody. And I really feel it's valuable. I appreciate the questions. And I want to make sure that we keep interacting. I think it's easy in these virtual environments to not interact. And I think your willingness to be engaged and ask questions is important. And I appreciate it. I hope people will continue to feel comfortable asking questions. And we can always pull up. I have a drawing tablet. I can always pull up a screen share of a whiteboard, and we can write on the whiteboard. So if we need to, we have the equivalent of a blackboard available to us as well. Haven't used that today or so far, but we could do that. And so I'd much rather take an extra lecture or two and make sure that everything is clear. And especially with the homeworks, especially when we do the homeworks, with the homeworks given a little bit ahead of the class, the motivation for that is that when we cover it in class, you have questions. Don't worry so much. If you hand in a homework assignment, you don't get perfect score on every problem. It's all right. It's not the end of the world. The point of the homeworks and the point of the homework is also not to torture you. If you work on it for an hour and it doesn't make sense, don't kill yourself on it. Make an effort, see what you can learn, and if there's a problem, flag it and we discuss it together. And I realize it's not quite the normal structure of a lecture course, but I think I know in my own experience of doing this, I learned by doing it. I make mistakes. And I'm quite happy if Giuliano catches my mistake, and I'm quite happy if you catch my mistakes, or if I do a solution to a problem in an inelegant way, and Giuliano or someone else uses Numpy and does it in a more elegant way, that's fine too. I can learn too. I've used this code for a long time, but I use it in the way I'm used to it. It doesn't mean it's the best way.